Hey everyone, and welcome back to today's Explainer. If you've ever found yourself wondering, what actually happens on a molecular level when we try to reverse the aging process, well, you are in luck because we've got some absolutely incredible data to unpack today. We're going to dive deep into the science of how young plasma factors can literally unlock systemic tissue regeneration. It is fascinating stuff.
So here's our roadmap for the Explainer today. We'll start with the parabasis regeneration model. Look at activating skeletal muscle repair, protecting immune stem cells, and the dual mechanisms of rejuvenation. Then we'll tackle the tissue penetrance challenge and wrap up with how scientists measure real-world delivery. All right, let's get into it.
Okay, let's start by looking at the chronic parabasis model. Now, if you're familiar with the field, you know this is the model where the circulatory systems of two subjects are physically linked together. And what it proves is honestly remarkable. It shows that circulating factors in young blood can actually reverse aging markers, rebuild tissue architecture, and completely restore homeostasis in aged organs. This isn't just a theory on a whiteboard. We're talking about the observable physical remodeling of tissue.
Now, when we talk about systemic regeneration through these young plasma factors, it's definitely not a one-size-fits-all mechanism. Actually, tissue rejuvenation relies on a dual-pronged approach. On the one hand, it's aggressively activating systems to build themselves up. But on the other hand, it involves a highly targeted suppression of inflammatory senescence in the cellular microenvironment. It's a delicate balancing act. To see how this actually plays out, let's map out the targets. We've got skeletal muscle for structural rebuilding and the hematopoietic system for immune homeostasis. But there's a really notable exception here that I want to point out. While skeletal muscle and hematopoietic stem cells, or HSCs, are profoundly impacted by these young plasma factors, T cells in the broader lymphatic system show literally no direct alteration in this specific model. So it's highly targeted. Let's kick things off with this skeletal muscle story. I want you to think of this like slamming your foot on the gas pedal. Young plasma factors wake up dormant progenitor cells, specifically satellite cells, and drive them through this massive three-phase cascade of repair. Phase 1 is satellite cell proliferation, basically expanding the pool. Phase 2 is myogenic differentiation, turning those progenitors into mature cells. And Phase 3 is the actual structural tissue repair and extracellular matrix remodeling. But before we zoom inside the cell to see the mechanics of how that happens, we've got to recognize the systemic drivers. It all starts in the plasma. Circulating hormones and factors like testosterone, LAF, and ADM act as the critical initial triggers. Testosterone, which obviously circulates in high concentrations in young blood, acts as the direct driver for satellite cell proliferation. Meanwhile, factors like LIF and ADM push vital IGF-1 and Notch signaling pathways. So the really crucial point is how these extracellular signals actually change cell behavior. Those circulating factors cross over from the intravascular space, they bind to cellular receptors, and then they converge inside the cell to activate PGC1-alpha. This is the master intracellular switch. PGC1-alpha essentially serves as the ultimate engine for robust downstream myogenic activity. But regeneration isn't just about stepping on the gas, right? It's also about cutting the brakes. Specific intracellular factors actually have to actively block chronic inflammation pathways. Because without that blockade, inflammation would just halt the entire repair process in its tracks. By suppressing it, primary transcription factors are successfully mobilized so that cell differentiation can actually happen. And once those new cells are built, integrating them into the broader microenvironment requires three specific things. First, cell-to-ECM adhesion. You've got to physically anchor the new cells. Second, neural reinnervation, which is driven by factors like GDF5 to restore functional electrical movement. And third, angiogenesis, basically establishing new blood flow to feed the really high metabolic cost of this newly generated tissue. It's the whole shebang.
Okay, shifting our focus over to the immune system, we first need to understand the primary enemy of immune homeostasis here, SASP, or the senescence-associated secretory phenotype. This is basically a state where biological aging drives cells to essentially catch fire. They start secreting these incredibly high levels of inflammatory cytokines. And this chronic inflammation just paralyzes normal immune function and triggers cell cycle arrest.
So unlike the muscle's active structural build, commune rejuvenation is an active defense. We are trying to protect those hematopoietic stem cells, HSCs, from the threat of SASP. Young blood factors like GCSF, SCF, and IL-10 directly target the HSC niche.
The primary mechanism here relies overwhelmingly on just putting out that inflammatory fire and inhibiting those destructive age-related cascades. And the molecular blockade here is just fascinating to look at. I mean, how exactly do we dismantle SASP? Well, young plasma factors block the transcription factors that are responsible for unleashing heavy inflammatory cytokines. They also inhibit the signals that cause cell cycle arrest, and, get this, they neutralize the pathological unspooling of compact DNA, which is what allows senescence genes to be expressed in the first place.
On top of that, there is an incredible epigenetic and mitochondrial rescue going on. Young blood factors suppress YY1 transcription activity, and that prevents the downstream inhibition of essential pathways. By unblocking those pathways, mitochondrial metabolic function is restored. And the direct result of waking up those mitochondria? It completely rescues the motility of natural killer cells and restores the baseline proliferation capacity of our HSCs.
So just to quickly recap and compare our two main stories before we move on, we have an aggressive, active waking protocol for skeletal muscle on one hand, focusing on proliferation and architectural remodeling. And on the other hand, we have this defensive, suppressive protocol for the immune system, focused squarely on inhibiting senescence and blocking that highly toxic SASP environment. Looking at the big picture here, tissue rejuvenation relies on completely different master regulators depending on the biological dimension you're looking at. The key circulating drivers and master regulators are totally distinct for muscle versus the hematopoietic system. It really just highlights how context-dependent young plasma acts inside the body. Now for the reality check, because knowing the genetic pathways is one thing, right? But how do these massive circulating proteins actually reach the intracellular space of a deep tissue organ? This represents a massive translational gap in our understanding of how this actually works in a living, breathing organism. Honestly, this part completely blew my mind.
The physiological reality of parabiosis is that systemic rejuvenation is not instantaneous. No way. We're dealing with large circulating proteins here, like LIF or GCSF. They have to physically navigate and equilibrate across completely distinct, fluid-filled biological compartments over time just to get from the bloodstream into the actual organ tissue. Just consider the sheer scale of the physiological challenge through this three-compartment delivery model. Systemic factors are introduced into the roughly 3-liter intravascular space of the blood. From there, these plasma proteins have to slowly diffuse out of the blood and equilibrate into the massive 12-liter interstitial fluid, which is the tissue space surrounding our organs. Only then can they reach their final destination, binding to the intracellular space to enact all those genetic changes we just talked about. I mean, a 3-liter volume basically has to effectively control a 12-liter transit zone.
So how do we actually know they're making it across? Well, to measure this real-world delivery, researchers use this incredible technique called in vivo microdialysis. They literally insert a semi-permeable microdialysis capillary directly into the tissue space of a live subject. This lets them continuously sample the interstitial fluid, quantifying the exact concentration and diffusion rate of systemic factors as they arrive at the tissue microenvironment. It beautifully bridges the gap between theoretical biology and measurable pharmacokinetics. Pulling it all together, the chronic parabesis model really teaches us 3 core principles of regeneration. Number 1, it's a vast, interconnected network of systemic factors. There's no single magic bullet protein for aging. Number 2, young plasma acts contextually through tissue-specific protocols, building muscle while actively suppressing immune senescence. And number 3, the penetrance imperative. Any therapeutic application depends entirely on successful physiological transport across those fluid compartments.
Which leaves us with a final, pretty provocative thought to consider today. We understand the pathways, we know the targets, and we're actively developing tools like microdialysis to track the journey. So, if we can master measuring and actively improving tissue penetrance, how close are we to bridging the gap between cell biology and physiological delivery? Answering that question could literally unlock the future of anti-aging medicine in the clinic. Thanks so much for joining me on this explainer. Keep asking those big questions, and I'll catch you next time.